Hi, everyone. We're going to give a couple of seconds for everyone to come into the room. I just opened it up. Welcome to the Canatech NR webinar together with Dedi Meiri and the guys from Front Range Biosciences, Dr. Reggie Gardino and Dr. John Voigt. I'm the only one without a PhD at the webinar. I hope that I can keep up with all the scientists. For those that don't know, Uri is at NRGene. NRGene is a company that he'll tell you about, but is involved in the genomics and breeding of cannabis using computational genomics and advanced tools. Professor Meiri, everyone should know, is a professor of biology and botany at the Technion, is in advanced cancer research and is very, very well known in the international scene. Dr. Reggie Gardino from Front Range Biosciences is another PhD in plant sciences and genomics. And John, who joins us, is the co-founder of Front Range Biosciences and believes in the importance of sustainable agriculture. So we've got this panel together to really deep dive into how some of the advanced genomics tools can be used in real life situations. And with that, I'd like to hand it off straight to Uri, who's here in Israel with us and joins you to tell you about NRGene and some specific test cases that can show you the applications of how we use these advanced tools in real life situations. Uri, welcome.
Thank you, Sol, for the introduction. Can we switch to the slideshow?
Yep, coming up.
So good morning, good evening, good afternoon, all of you that are that have joined us. Thank you for joining this really special event. I'm glad that we have an opportunity to meet really three groups of speakers, or three speakers from all ends of the industry, so to call it. How do I switch slides here? Okay. Oh, okay. So I have a slide here that shows pictures of chili peppers, and you're probably wondering, what would chili peppers do on a cannabis webinar? So I've actually put this to demonstrate the far extent that breeding has taken chili pepper. It's a fairly new crop, and it's reached this amazing diversity of shapes, forms, and qualities, rather analogous to cannabis, and also the uses for food, for colorants, for all sorts of medicinal uses, and capsaicin extraction. All of these have been enhanced and improved through breeding. And in a lot of ways, it's very much analogous to cannabis, multiple uses and a lot of applications on the medical side, on the agricultural side, and lots of products. And all of them could be improved and need to be improved through breeding. And this is the context of why we're all gathered here, to show you some real life examples and demonstrations of how breeding take us where, okay, so there's a delay in switching slides, sorry about that. Okay, so a few words about NRGene. We are not a cannabis company. We are a genomics company. We're based out of Israel. We've been around here since, I think, for the last decade. And we do genomics in many crops. Most of our business is in the seed industry with big seed companies, and also finished products of paper, beverages, and whatever. And only about a third of what we do is related to cannabis and hemp, but we've already taken a strong and significant presence in this cannabis and hemp space, mostly with large LPs in Canada and large companies in the US, and also European and Israeli companies. There are about 40 employees, mostly in Israel, but also in San Diego and Saskatoon. And we have established technology, and we do the service as a genomics computational analyses for others. We are not breeding for ourselves or generating our own varieties. Okay. So the overview of how we handle breeding is to offer solutions that simplify variety development and gene discovery, meaning we go to the client, we analyze their large-scale breeding populations, their collections, we help determine the right combinations of parents. And then we create with the client large-scale breeding populations and analyze those computationally. We sequence and analyze the data, and we help make breeding decisions and advance research using these. Well, there's how do I? Okay, there's a note that blocked my screen. Oh, could you try to go to the previous slide? Okay, so we help the clients make breeding calls, breeding decisions based on molecular markers. And the end goal would be to develop new varieties and genetic information for research, for IP and development of varieties, either through clones in the short term. And the end goal should be for seed production, which is, we think, would be a more efficient use of producing cannabis, especially in the hemp space, where the major advantages of how we do this type of breeding is the low number of plants that we need to do this R&D project and development of marker and small space required. We do it very fast. Developing of a new variety in the past could have taken over 7 years. We do it in around 2 years. And there's no need for phenotyping the entire population. Once we get genetic markers, we move to breeding only with the genetic markers. And obviously, we do this on the client's germplasm. Is it? Right, it takes quite a while for the slides to change, so excuse me for the delay. So most of the breeding challenges that we receive from our clients revolve around productivity and common traits in the cannabis and hemp space. I would say the #1 request would be powdery mildew resistance and botrytis resistance, because this is probably the largest pain point for cannabis and hemp growers. Then the next step, the next target for traits and requests are usually around yield. So yield is a complex and compound trait that could be broken down into several smaller traits that are easily analyzed and could be selected for, and markers for the specific positive traits could be selected for. For example, bud structure or agronomical performance, flowering time, etc. All of these combined are components of what people call yield. And then the other common requests are specific cannabinoids and either major cannabinoids, higher concentrations, zero concentrations in the case of hemp. And to some extent, there is desire to control the terpene profile, which is a much more complicated trait to control genetically. But these are the common requests. And then there are some cases where we have an odd request for something more exotic. And this is one of these examples that I will show today. Yes. So the example, and this is a real life project that we are working on. There's a variety coming from Hawaii that is called Blood. If you have a large collection, you may have seen examples of this phenotype. So this produces a very strong dark red resin or sap in the stems when the plants are fully mature. And This particular sap is not only cool, but also has medicinal traits or associated with the medicinal traits. People who've consumed the flower report numbing of the peripheral nerves, sometimes the arms and feet, and mostly they report numbing of the face. So this is a super important lead for us. This is a trait that we would like to isolate, explore, research, and transfer from one variety to other varieties that are better producers and have other desirable traits. So this is an actual F1 population. The way we work is, as I mentioned in the overview, our clients produce a segregating populations of about 200 individuals. As is, we take the F1 population. If the trait segregates, and in this case, you can see by the pictures, they segregate very nicely from extreme red to almost transparent sap. And each variety of, or each line or each plant of the 200 individuals will have a different genetic composition. As seen on the bottom lines or illustrated on the bottom lines. From the same original combinations, you have recombinants and different combinations of the genetic makeup that make this. And our job is to isolate the regions in the genome that are responsible for this phenotype or this red sap trait. So the way we do this is, first of all, the client produces the segregating populations, and we look to see that the trait actually segregates. If it doesn't segregate on the F1 or on the F2, or you cannot measure it, it's something obscure or not measurable, then this is not a mappable trait. But all other traits, no matter if it's the first time we've ever seen it, we can map it. So we need to be able to measure in this case, it would be the color intensity in RGB of the sap. The next steps I'll elaborate on digging deeper into the compounds, but we would look at the intensity first and correlate specific parts of the genome with the strong and dark red color, eventually discovering the region in the genome that is responsible for controlling this trait. We call this quick genetics package. So you can map within this population, this trait, and many others that segregate in the same population. And then we could also take the region of interest. Hold on, it went back to the next slide. Okay, no, the other backwards. Thank you. So we zoom into, and computationally, we could zoom into the region of interest and compare it to our Cannagene database, which is a vast database of genomic information, and compare the content of this particular variety to the equivalent regions in other varieties. And even without prior knowledge of what we should expect there, we could produce genetic markers that would enable to transfer this trait to other varieties. Okay, next, please. Okay, so the Cannagene database allows us to computationally define a region of interest. We did this by the QTL mapping. We've defined the region associated with the trait. So now we could zoom into the region of interest and compare this particular genome to over 15 genomes that we have in the database. And we could look at regulatory elements, present absence variations, copy number variations, and new alleles or novel genes. In this case, if there are new genes that control this trait, we would see interesting new regions that do not exist in other genomes. So the next steps, obviously this is not the full picture. This defines the region of interest and brings us to the next steps. So the next steps is usually collaborations with other research. In this case, this is Dedif, and who will talk about this in great detail later, or projects like this. And we need to identify the red compounds, whether it's a flavonoid, a canflavin, or a novel compound unknown. And then we could isolate the compound and test it in model systems, cell system receptor interactions, live models, and we could test the synergism of this compound with cannabinoids and hopefully reach new leads for pharma industry and medicinal cannabis to isolate compounds that affect pain perception and the numbing of the peripheral nerve. With that, we'll bring this topic to Dedi. He will elaborate a lot more on the next step. Thank you.
Hi, everyone. Thank you for being with us. Thank you very much for Ori. I will take it from here. When we see a phenotype, a plant that is different from any reason that we think something is different, we don't know where it's taking us. And in order to do so, we first of all, usually analyzing the plant So we know that the plants have cannabinoids. So we first of all analyzing the full spectrum of cannabinoids. We can analyze today a kind of variety of around 140 some cannabinoids. Usually in one plant, you see much less. There are around 10 families of cannabinoids, and we can analyze them. We can analyze all of them. To part of them, we have standards. Part of them, we develop a library that can, in a high percentage, we can assume which cannabinoids it is. In the same way, we can do to the terpenes and the terpenoids, and the same thing for flavonoids. So we can analyze around more than 100 terpenes, 100 cannabinoids, around 40 flavonoids, and other compounds that in the plant. So first of all, we develop a methods to analyze what we think is the active compounds in the cannabis. I must say that when you run a LCMS or GCMS on resin, on flowers, on extract, you get hundreds and thousands of picks. We are focusing on the cannabinoids, terpenoids, and flavonoids, but generally there are many more compounds that usually we are ignoring from them. But if they will be responsible for the effect, and I will show in a few seconds how we narrow it down, we will examine that, and there is tools to examine compounds even if they are not known or a new one, using NMR and other devices in order to learn of them. And we already analyzing compounds from the cannabis and from other plants that we're working that is unknown compounds, and we start trying to learn what they're doing. So these are the terpenes. Ori was right that changing figures here is very it's not easy. I'm pressing before I'm starting to talk to change the slide and waiting a lot. So when we have this kind of a plant, first we are analyzing all the active compounds that we assume are there. And then we will take it to what we call high throughput screening. We will screen this on many biological methods and biological systems that we think this one or this plant can affect. We have equipment, we have robots, and we have equipment to screen many different types of, in different methods, usually cell lines and animal models. And when the slide will move, I can show that. So we developed cell-based for disease models to understand the biological effects of many cell-based diseases. We are looking on lung diseases. We are looking on neuronal diseases like Alzheimer's, sleep disorder, multiple sclerosis. We're looking on inflammation, how it's affecting the immune system. We developed a variety, a bank of cell models for diseases in order to check when we're starting from something that we don't know what it is. We're doing it, as I said, in a high throughput screening. Usually we're starting very fast with a fast screen to get an idea, to get the clue, to get the tip of the rope, how it's affected. And it can go for every type of how it's affect the cells, whether it's affect their divisions, their differentiation, how it affects the molecule pathway, how it affects our immune system. Usually, I don't know where I'm starting when I'm starting with a new plant or a new kind of cannabis. And again, it's true from algae from the sea or a mushrooms or everything that I want to check. So after we are defining kind of a phenotype, something that this plant material is affecting, whether it's affecting the calcium release from the neuronal, the receptors expression, whatever, something that I can put my finger that this extract and this plant can affect, then I'm starting to ask, what is the single compound or combination of compound in this plant that's doing this effect? The way that we're doing it, we're taking the whole extract and we're starting to fractionate it. We're starting to cut it. We have equipment for that, whether it's HPLC preparative, CPC, and other machines that can do these fractions. I wish the slide will move. Okay. Let's try to move it. So these are type of cell models, and what you're getting in the end, it's kind of this kind of graph in where it's working and how it's affecting, and I'm trying to go ahead. And when you look on the compounds, it looks like that. This is from HPLC preparative, and we started to fraction. We're looking at that, and we see that there is a lot of compounds, and we're cutting them to pieces. In this case, we cut them to 4 pieces. Let's say we have 100 compounds. Now we cut it to 25 in every vial, and we're going back to the cells or to the mice model. And when we know what is the phenotype that we're looking for, we see whether one fraction is doing that effect. Sometimes we are lucky, like in this example, that fraction 2 work as good as the whole extract, and sometimes we need a combination or different fraction. But after we have that, we're going and refractionate the fraction that work, and we're going ahead and doing that more and more until we reach to the minimal compounds that doing the effect. Sometimes it's 1 compound, sometimes like in this case that I'm showing here, it's 3 compounds that when you combine them together, you're getting the activity of the whole plant. You can see in the left bottom that every combination of each one of them, of these compounds, C1, C2, and C5, these are 3 cannabinoids, didn't work, but just when we put all the 3 together, we get the whole action. After we're doing that, in order to prove that, first of all, we learn about that sometimes it's a unique compound, like in this sample, we had a unique cannabinoid that we wasn't aware about that before, and then we analyzed it, we purified it, we did an NMR, we learned on the structure, and then we can synthesize it and have it synthetic and check whether the synthetic is working as good as the plant one. When we have this one, we're going back with these 3 single compounds. In this case, it's 3 single compounds, and we're going back to the mice model or to the cell model and checking whether these specific molecules is working as the whole plant. We can do it to cannabis, and we're doing that to cannabis, but as I said, it's not just cannabis. My lab is working more than just on cannabis. I have a PhD student that collects 96 different algae from the Mediterranean Sea. We took them, we did extraction, we isolate compounds from there that affecting different diseases. We're starting to work with different types of mushrooms now in the lab and see how they affect. It's actually, I think that the cannabis opened the door for complicated biology with complicated plants that usually in the past we were afraid to touch them, and the pharma company and the scientists didn't start to work with them because of the complexity. But solving this in cannabis opened the door to do it with other plants. this is my lab that's doing this amazing work. These are my students. But on top of that, I have a company that's called Cannabis that giving me all the analytical tools, separation, isolation, and a lot of other things that I'm using. And this is the team in Cannabis that I need to say big thanks to my lab and to them. That was me, and I guess we are going ahead.
Yes. Can you specifically, Dedi, for a minute, just talk about the Red SAP project and how the clinical applications for that are working out, to tie it back into
Yes.
Uri?
So this plant showing a kind of a neuronal effect on patient, and we will focus on that aspect. We have in my lab, first of all, an animal model to a lot of neuronal diseases like multiple sclerosis, Alzheimer's, sleep disorder, and others. But in this one, we are checking the neuron activity. Whether you're checking calcium, you're checking potential of the neuron, and we have all these kind of cell models and animal models. And the idea is, first of all, to analyze all the active compounds in this plant and to see if they're a unique compound that usually we don't see. And in parallel, to check it on these cell models and the animal model to define an effect, and then to try to narrow down which compounds in these plants are important and doing this effect. The red phenotype is just giving us a hint that this plant is unique. I think what is unique about that is how it affects the patient, and actually the severe effect that they are reporting. It's not a good one to be paralyzed, but it's telling us that there is something very strong there that affects the patient. For me as a cell biologist, and the meaning it will bind the receptors, doing kind of activation on the cells, and that's what I'm looking for.
Amazing. So that is an example of how a trait in the plant has been taken more to the clinical side. I want to jump to John Voigt, who will tell us a little bit about Front Range Biosciences and how some of these traits are affecting the plant growing cycle. John, over to you.
Great, thank you. And thank you, Didi and Ori, for the informative presentation there. That was great. So I'm just going to talk for a minute just to introduce Front Range and Reggie, and talk a little bit about what we're doing and our perspective. You can actually go ahead to the next slide. Front Range, we're a little bit different than NRGene. We use a lot of the same technologies and are doing a lot of similar research. But we actually have our own team of breeders, and our team is led by Dr. Reggie Godino, who you'll hear from in a second. And we're primarily focused on the development of new cannabis varieties that have a broad range of traits that can perform in different climates all over the world. This crop represents an enormous opportunity for a broad range of industries, as we believe the ingredients that can be produced from this plant include not only cannabinoids and terpenes, which are the primary focus right now, but there's other small molecules in this plant as well. It's also got a balanced protein source with unique nutritional benefits. It's got a balanced amino acid profile, fatty acids, omega-6, omega-3s, dietary fiber, and it also has fiber that's used in industrial applications. The market right now is primarily dominated by cannabinoid ingredients, but as the supply chain develops, we anticipate significant market growth in some of these other areas as well, all leveraging this incredible plant. We also have a program in coffee, but we're not going to talk about our plant breeding and nursery work in coffee today, as the topic is cannabis. Next slide. Thank you. So this is just an overview slide of the scale of this industry currently from the agricultural perspective. And at Front Range, this is really, how we view this crop. As I mentioned, we see it as a source for lots of different ingredients. And the challenge is I had to put this slide together with some estimates and models. So these are all estimated, whereas compared to corn, you have USDA data and other governments report on it as well. But this gives you a sense, and this is all some estimates based on the market from last year with hemp as well as regulated cannabis. I think it really shows the potential, but it also shows that we're still pretty far behind corn in a lot of ways. And I think the technology is catching up, the industry is scaling up. But if you look at where corn is compared to where cannabis is, this is still a fairly small crop in terms of total acreage around the world. So I'm going to turn it over to Reggie here, but as you're going to learn from him, we use a lot of the same technologies and actually similar to some of the research that Didi's group is doing. We also have a chemistry group that's looking at novel chemistries and interesting new cannabinoids in this plant as well. So we're excited to be here, and I'll turn it over to Reggie, who's going to walk through a case study on one of the traits that we're looking at from an agronomic perspective.
Reggie, you're on mute.
Hi, everyone. Are you going to control the slides there? Because I don't see the control. So I'm just going to I'll just say change slide there. How's that work? All right,
Yeah.
cool. All right. So hi, everyone. I'm Reggie Godino. I'm the VP of R&D at Front Range Biosciences. Next slide, please. Today we're going to, as John said, we do a lot of the same work that NRGene does. And so we use the same kind of marker-assisted breeding, the same sequencing. We do a lot of expression analysis. The difference being that one of our goals is to develop varieties and markers at the same time. And that's not usually done because typically it requires a lot of space and a lot of germplasm to be able to do something like that. Which we don't have either. But by the proper approach and combining the right kind of methodologies and brute forcing, you can actually get to the point where you can develop markers in a rapid time frame and get product to market at the same time. So what I'm going to present is a case study that came to me kind of over this past winter. I have only been, me and my team have only been with Front Range since the beginning of August 2019. And sometime in October or November, I saw this video, or I saw a report of this video. And so we'll just, I'll use the video to set up the—
Colorado in 2019, we got hit by some snowstorms and some hail and some crazy amounts of rain. We thought for sure those first plants were going to die. And I remember looking at the forecast, and I was like, oh my God, we are going to get hit with a foot of snow. And it buried them. And we went back out there, and it was like they looked dead. But then, if you looked closer, you saw that there was new growth. 2 weeks later, blowing up, and I'm like, they all rebounded with very little sign of any kind of damaging, which was unbelievable.
Thank you. So as a little background, so Front Range believes in field trials. We do extensive field trials. We do field trials through partners. We have our own field trial program. A lot of that data that we collect in the field is very important to what we do, right? So when I saw this video, I was like, wait, we have a frost and snow resistant lineage? Like, how come I didn't know about that? So we immediately took that and started to breed it. And here are just some of the conditions that we dealt with last year. We had 3 varieties in that particular field. Over the course of 2019, those plants saw 3 freeze events that got to 27 degrees Fahrenheit or -3°C or lower, and 4 snowfalls, one of which was very early. You saw the field that he was talking about, that was right after planting, about a few days to a week after planting, and they were buried in a foot of snow. So our observations for the three lineages that were in that field were that one of them had no damage, the one he was talking about, some had minor damage, and then some had heavy damage. So the step one of the project was to identify the trait in the breeding population, which fortunately for us, his field gave us all in one fell swoop. Next slide, please. So the next thing that we needed to do was to determine kind of what our strategy was going to be, right? So we needed to be able to do rapid identification, minimum number of crosses to get to the genetic information. And we absolutely needed to develop the cultivar and marker at the same time, because now we have customers who are like, wait a second, we're in the northern latitudes, we want this right away. So we needed to be able to develop the cultivar and marker at the same time. And we had these three phenotypes that we could see. I say genotypes in question mark because we don't know how many genotypes there really are. And we had a spectrum of response, right? So the next step was to establish our mapping population method. Next slide, please. And this is where it kind of got, oh, sorry. And so this is actually the end result. So this is the same farmer you can see here on the right. These are the fields that he was talking about. So we did actually get, for this particular variety, a very good harvest from that field. Next slide, please. So this is where the problem came in, is given the fact that we had these three varieties, we potentially had mapping to be able to build mapping populations, but mapping populations are typically built with just two varieties. And I didn't want to lose the potential genetics, so I didn't know how we were going to, how those things were going to segregate. So we chose a hybrid approach, and I've listed here, you know, some of the different kind of approaches to building mapping populations. There's F2, you know, F2 normal kind of F1 self-cross or intercross between males and females, a back crossing to a specific parent. They each have their advantages and disadvantages. Recombinant inbred lines are really not useful at this point because that takes many generations and that doesn't hit the need part of getting it done quickly. Nested association mapping and magic are two other ways that use a lot of the molecular diagnostics that both Ori talked about before and that we use. And so we developed a hybrid approach, which is actually a combination of two of the different mapping approaches. And we combined that with DNA and RNA sequencing and genome-wide association. And then so on top of that, we had to further modify the methods because our mapping population did not fit the classical definitions, because for a magic mapping population, you need 8 parents. We didn't have that. We had 3, right? So, and so we really had to kind of think about how to combine the best parts of a couple of different methods to be able to get what we wanted and meet the time frame that we were looking at. In the downtime, while these things are going on, we did a lot of bioinformatics to look for paralogous genes in other plants. That's a lot of the kind of stuff that Ori was talking about as well. We actually are going to use stressing and RNA-seq to get a lot of the information. So part of this will be to grow the plants under these cold or near freezing conditions to see whether or not we can elicit that effect in the RNA-seq analysis. And then we took it one step further, and we have recently put online a predictive breeding software called Phenome One. It comes from Israel. It's a phenomenal piece of software that we have embraced wholeheartedly. It is allowing us to track all of our data and to actually follow lineages and make some kind of downstream analysis or guesses as to what we should be breeding. Next slide, please. So the next step was then to actually start to do the crosses. So we actually have, at this point, the stuff in black is what we're currently doing. So we actually have seizes from the multiple F1 populations that were developed. They are all currently right now being grown vegetatively, being prepared to be put into the growth chambers and stressed so we can get the RNA-seq analysis. That's all going on right now. While we're getting this data, we are also using the Phenome One software to help predict what outcomes should be. And all of these things through the vegetative propagation of these F1s going out into our field trials right now in the northern latitude locations. The next step, which is in blue, is for us to take the data from what we get from both the cold stress and the field trials, do further RNA-seq and bioinformatics to kind of narrow down the markers that segregate with the population. Next slide, please. So our hope at this point is that the combination of these two things will give us and the bioinformatics for paralogous genes from other organisms where we know there are some cold resistance phenotypes, is that we'll be able to use that information to get to a place rapidly where the field trial data and the RNA-seq data comes together to be able to pinpoint the QTLs necessary, as you know, Ori pointed out, the QTL analysis and getting to the meat of the information, the actual marker itself is the part that we need to get to rapidly to be able to then turn this into a stabilized population, right? So along those lines, we're using another piece of software called AgriTask that comes from Israel as well. It is our choice for our field trial management. It allows us to build apps and everything else that we can go in field, do our data collection, all of our phenotypic analysis, picture taking, all within, and it all gets dumped into the same database. And we have our Pheno1 and AgriTask database talking to each other so that we can cross-correlate that information. Very important for big data management. And so again, I say step 5. So at this point, we will hopefully have the marker identified, and we will, while having developed the variety, because these things are all being vegetatively propagated and put out into the field trials. Next slide, please. Next slide. Hello. Oh wait, something happened there. Right, okay, cool. So the results are, after the screening, that we will have cultivars already in our customers' hands because of the vegetative propagation aspect. Our field trial data will allow us to have moved towards the final candidate selection that will be that large-scale vegetative propagation release. And at that point, those vegetatively propagated mothers, right, will then be the next line for the F2s and recombinant inbred lines that will need to be developed to stabilize that to offer it as seed. But in this scenario, having started just around January or February with that first set of crosses, we do expect that by September 2021, we should have a vegetatively propagated line in the field that is resistant, and shortly thereafter, a recombinant inbred line that is stabilized for seed. And that's it. Thank you very much.
Thank you, everyone. That's very, very informative. I have a quick question from Dr. Russo. Thank you, Ethan, for joining us. About the red sap, Uri, has this been published anywhere?
Not yet. This is a work in progress. We are now, what I've shown you is the phenotypes. Those have been quantified and genotyped. So we are now at the analysis stage and passing samples to Dedi's lab for analysis. So this is going to be published eventually, but not yet. We're not done.
Ethan, it's a great honor to have you here. I will be very happy to publish it in Cell or in Nature and Science, but we need a few years and then few results for them.
So I've got a question for each of the panelists. What are some of the most interesting traits? You've shown us red sap, you've shown us a hardy frost strain. What other things are there that you're looking for or that you've found? Uri?
So most of the projects we do for others, so I'm not at great liberty to discuss. I think I mentioned the most popular ones. First of all, we do have a motto. If you can measure it accurately and it segregates in the population, we can map it.
Yeah.
That's the general rule. Specifically, as I've shown, powdery mildew is probably the hardest demand. After that, people want autoflower for indoor growth and controlling getting another crop cycle. And then you have the agricultural performance, higher cannabinoid, lower cannabinoids, no THC in hemp, high THC in medicinal or recreational. And obviously, there is interest in special terpenes. So those are also requested. And then straightforward agricultural traits like branching, like different plant architecture traits. So all of the things that, and I think also Front Range guys, Reggie and John, demonstrated this. This is a classic agricultural breeding trait, frost hardiness, very straightforward.
Reggie, some of the interesting traits besides frost that you're looking at?
So I'd say, we're, I agree with Ori. If you can measure it and it segregates, it's a target for breeding, right? So we're obviously interested in the minor cannabinoids, right? So we're doing a lot of work with CBG, with varins. We're bringing our chemistry. So at one point I came from a lab that was one of the top labs in measuring chemistry, and now it's Dedi's lab, and I'm behind, and so I have to catch up. But we're approaching it the same way. So we're really looking at the various chemical groups, not only the cannabinoids and terpenes, flavonoids, all the other biologically relevant compounds we know from other plants, right? We know that there are, antioxidants. That all of these things that we see in other plants exist in cannabis to a certain degree. So those are things that we're interested in as well. Agronomic traits, because, ultimately our customers have to be successful. So one of the things that I have taken on since we've gotten to FRB is a better understanding of these agronomic traits. Branching structure, how well it holds up to wind is related to branching structure. Some of those things are very important. So I would categorize regional performance as another broad category of things that we're looking at. And then, as John mentioned as well, we are also, because we are a full spectrum hemp company, to borrow the word full spectrum, we also look at grain and fiber. So we have discovery programs going on in both of those as well. We're looking at how to, improve tensile strength of fiber hemp, to improve the quality of that fiber, right? So there are some fiber varieties around the world that are known for their high quality fiber, and others that are not known for their high quality fiber. So things like that. And again, the nutrient factor as well. Hemp is one of the best protein sources on the planet. Pound for pound, it is as nutritional as beef, lamb, any other meat, right? So and it costs 100-fold less per acre to grow. So we're very interested in that market as well, to try to look at producing higher quality hemp and or more seed per plant per acre. So
Dedi,
Yeah,
that are particularly interesting.
few things that I saw previously, which are very interesting. There is a strain, high CBD strain, that its CBD doesn't act as the regular CBD. So when I purify the CBD, it's not acting like the other CBD. It's not producing seizures in the mice. It's not affecting as other CBD. And we're trying to understand why this CBD is different. And this is very unique things, something that high CBD strain, which all the downstream family compounds look like CBD. It looks like CBD in the mass spec, but it's actually something there is different. We purified it, we send it to inspection, and I don't know still what is the answer, but this is something that is very interesting. We are looking for unique cannabinoids and unique flavonoids, and there are a few strains that produce them, which is very interesting for me. One strain that I saw a few months ago that it's extremely interesting, it's a strain with 17% CBG, 0 THC, 0 CBD. And what is interesting about it, that you can see the pathways of the cannabinoids, which cannabinoids disappear and which cannabinoids is still there. and I think we can learn a few things about the pathways of the terpenoids and cannabinoids from this strain. It's extremely interesting, even though this is more Ori and the Rigi part of this puzzle. My PhD in genetic plant biology, so it's interesting me as a scientist. I'm not doing much with it, but this is very interesting. I just want to say one comment to Rigi, which is a very good friend of mine. Two things. First of all, I saw that you're using a lot of programs from Israel. You're bringing a lot of programs from Israel. I think it's better for you to come to Israel instead of bringing them. This is first thing. Second thing, I'm not sure that the freezing strain that you find will be bestseller here.
It will
You don't have
grow.
mountains? You don't have mountains?
We have snow for a
I
week.
have a politic answer for that, and I won't say it here.
All right. So Denny, you stole this from me. I was gonna say that I'm very happy that Front Range, and this was not coordinated, used two software tools developed in Israel. I actually know the people involved in at least one of the companies, probably the second as well. So also, we are trying to, not trying, I think in the last decade and a half, Israel has been implementing its expertise in software tools and bringing it to genomics and to developing tools for better breeding, faster and more efficient on all aspects of the process. And we're starting to see results. So we're very proud in that too.
My response to Daddy is, well, I tried for many years to move there, my friend, and you were involved in that, and it just never worked out. So this is the next best thing for me. But I will say, the important part here, I think, to wrap this all up, is the two groups that I'm using, right? Pheno1 and AgriTask are people that I met at Canatech conferences in Israel, right? So the background was forged through this educational series, right? So through the Canatech program, I met the people that I brought along with me into this project. So,
guys, there was a couple of other questions on here. There was a couple that were directed at us at FRB. I can answer those really fast if that's okay.
Please.
So the first question was about being focused on hemp varieties. Are we doing any work in THC? And the answer is yes. We have a breeding program out in California, working with a licensed nursery up. So we see cannabis as the whole plant, whether it's got high THC, low THC, and the hemp versus cannabis question is really just, or THC question is really a regulatory consideration from our perspective. So yes, we are breeding in that area. The other one was somebody asked about if the snow plants, or the snow resistant plants were grown from clones. Those were from clones, right. And they were photoperiod sensitive plants. And I don't know the exact age. Our typical age for rooted cutting is when it ships out to be planted is anywhere between 3 and 5 weeks on the oldest. So it just really depends on the plant variety and how fast it rooted. We have quality control metrics to make sure that it's ready to transplant. So those are the two questions that we had.
So let me add to that last piece. So what was their age at the planting, the first snow? So they were planted May 30th. The snow happened, they were planted May 21st to 23rd, and the snow happened on May 30th, right? So it was a late spring cold snap that the temperature got down to about 34, and they literally had 1 foot of snow dumped on them. But because of Colorado, the snow was pretty much gone in 2 to 3 days. So it wasn't a long lasting cold, but it got cold. They were buried for a couple days, and then it melted.
Shaul, I have a question for Ori and for Rigi. How many generation, how many F you need in order to stabilize really a strain? Like there is kind of a finger saying in Israel that you need F6. Is it right?
It depends on what your definition is, and so I'll let Ori give probably the more traditional answer. But you know, I think what people need to understand is are you concerned about the entire genome, or are you concerned about the traits that you're interested for, right? If you're concerned about the entire genome, an organism the size of cannabis would typically take somewhere at least 8 generations to get to about 90 to 92% homozygosity across the entire genome. But if you were to look in a field, that would still mean that 1 out of every 10 plants looks different, right? And you won't, and you could have multiple populations, right? So a true breeding strain, as defined by agriculture, is typically somewhere between 18 to 20 generations of inbreeding. So I'll let Ori take it from there.
Yeah, so first of all, I think cannabis prefers to be outcrossed, and it's very, very difficult to get a fully inbred cannabis lines, and there are no doubled haploids commercially viable technologies to do this quickly. So if you're trying to do inbreeding and stabilize the entire genome, first of all, you would get by F3, you may get very strong inbreeding depression. And besides that, there is no exact need to fully stabilize the entire genome. You need to stabilize good enough. So the rule of thumb on F6 is probably still valid. Maybe if you do it efficiently enough, you could do it much faster than F6, because not all segregants are created the same. If you analyze those and you have full genome markers throughout the entire genome, you could make shortcuts and reach higher levels of stability on the regions of interest way before F6. What I usually recommend to clients is to aim for seeds and stabilize the traits if you're going to go for seed production. But if on that way a good clone comes along, you propagate it and you sell it, and if you're lucky enough, by first or second generation, you have a commercial crop going on. So super fast on the way to seed production.
Yeah, and that's how we do it as well. That's why the case that I presented was we're developing the line and the markers, and we hope to have the entire process, you know, to the point where a commercial variety is ready to go in 18 months. So.
Can you talk a little bit about genetic drift and how that affects, you're talking 18, 20 months for 20 generations to stabilize the genetics. At what point do they start to shift, and how can you prevent that, Uri? By are there tools to prevent that?
So without going too much or too deep into genetics, because this is a very complex question to answer, I would just say that yes, there are tools to minimize genetic drift and to control genetic drift. There's a lot of questions on how you make your propagation. Are you going from clones, leaving from seeds now and then? If you talk to people who do propagation from clones, they might tell you that after 3 or 4 transfers, suddenly the plants change. So this is not exactly genetic drift, but this is losing your clone. This stuff happens. So if you're working with clones, the best advice would be to keep multiple copies ongoing and keep a good genetic record of what your germplasm should look like. So if you have a mix-up, or if you have a suspicion of some sort of drift, whether by crossing or by somatic mutations, you have a reference to look back to and decide, you know, this is where I want to go back to. And then if you're doing crosses and trying to establish something, if you have proper genetic markers throughout the entire genomes, you could always do another backcross and return to the genome of interest. You need to be able to discriminate though between what you want to keep and what you want to discard.
Any other questions from the panelists before we hand it to Uri to summarize? That would be no. Over to you, Uri.
Yeah, there have been a few questions addressed generally to the speakers about import, export of various clones, seeds, plant material across state lines, across country lines, and so on. So there's no one answer depending on what you want to transfer from where to where. Each country and state has its own regulations and rules. So shoot us an email with specific questions, and if we could help with problems in this area, we do. That's kind of a general answer on that. So I would like to give really great thank you to all the speakers, to Dedi, to Reggie, to John, and to Kay for Saul for moderating the webinar. I do like to summarize what we talked about today. I think we've shown really a diverse set of examples how we could move from ideas, research, problems in the field, challenges, interesting finds, and use molecular tools to research and develop quickly, relatively super quick in the plant world, new varieties, new products that either are ready to sell right now or have immediate sale as clones and seeds, and later on other uses as products in the medicinal and clinical line, which have obviously much greater value. At Energene, we help others. We are not developing our own varieties. Unlike Front Range, we do this, and so feel free to reach out to us and ask, and we'll be happy to provide details on what we could do for others in terms of breeding with your germplasm. So I do think that combining molecular markers shown here today, computational approach, biochemical research, all of these capabilities demonstrated here by the speakers delivers high value to generating new varieties to customers interested in developing their own breeding programs and their own research goals. I think it's a high value and high return on investment to develop new types of varieties. So again, please feel free to reach out to us in questions about this webinar. It will be available through the Canatech platform and at the websites of the companies, and I think Dedi's lab maybe. And so feel free to reach out with questions and project ideas, challenges for breeding. Thank you very much.
Thank you, everyone. I'll make everyone's emails available. If you have specific questions, you can always come through me or go directly. Thank you guys for joining us from the US, and Dedi, as always, the time. I know your time is precious, so we do appreciate it. And thank you for elevating the science around cannabis and breeding, and hopefully we start to see these applications in the patient population. And Dedi, you blew my mind that not all CBD is the same. We know not all strains are the same, but now it's starting to be even more complicated. So we look.
I just want to say, because Ethan Russo is on the line, and I'm afraid all CBD are the same. Probably what I see, it's not CBD.
There you go. But thank you, everyone. Thank you, John, for joining us, and Reggie. And we'll continue to see you weekly on our webinar series. Thank you.
Thank you very much, everybody. Bye-bye.
Thanks.
Great. Thank you.
Thank you. Okay.